All right, guys, so we have done the first video, the first section of chapter one, and then we are going to finish up the last section of chapter one. So the first one was properties of life and levels of organization and what is biology and a little bit of Earth's history. And then we are going to cover the second section of chapter one in this video, and that's processes of science, like how does science even work? When we are in today's day and age, science is so important, especially right now in the middle of a pandemic. When I'm filming this video, science is important. It is going to, the textbook says, gather knowledge about the natural world, and it has transformed human existence. You betcha. So many advances have been made because of science. We are living longer because of science. So many things. So let's talk about how it is always changing, always evolving, right? We talk about those properties of life that you needed to know, and being able to adapt to change and evolve is part of those properties of life. Science, too, is always changing, always evolving. We are always learning more. Um, of speaking of the pandemic and the COVID virus, um, this was is a brand new virus. We have seen coronavirus before. We have seen it coronavirus before. The common cold is a type of coronavirus. SARS uh, is a coronavirus. This is something that we've seen before, but we have not seen this strain before. So this is new. It's using it's a different um, genome inside with this one. It's evolving. Our knowledge is always evolving. We, at first, didn't know exactly how it spread, right? Was it just through direct contact? Was it through fluid transfer? All fluids? Does it need a blood transfer, or can it just transfer through respiratory droplets? We are learning more and more about it. How does it attach into your body? Where does it attack your body? What are the symptoms that are involved with the infection? Why are some people have these symptoms while others don't? We are always learning, and that's just coronavirus. We are constantly learning about everything. I mean, I'm always curious about what things are going on and why they're doing it. Because I love science. I think science is cool, and I hope you do too, or at least if you don't love it, which is okay because I don't love everything. I'm not a huge writer, English. Me personally, writing does not get me excited, but science does. And my English friends can write a stellar paper. I have some really good friends in the English department, and they rock in English, and they may not love science, and that is okay. But I can still respect, English, and the English majors can still respect the science. So we need to understand what's out there, even if, biology is not going to become your all consuming passion. That's okay. But we still need to understand what's going on with it and have a basic knowledge. So, again, science is always evolving. We have learned that there are some really old life forms out there. We learned in the previous video that the Earth was 4.6 billion years old, and the first forms of life showed up about 4 billion years old. And those guys were prokaryotes. They were going to be bacteria and archaea, and those guys colonized all the crazy places, the super salty lakes, the polar ice caps, the bottom of the ocean where there's no sunlight and they're only using chemicals for food. I mean, all the crazy places. The prokaryotes were there. They came first, they adapted, and they were able to spread everywhere, which is super cool. So they are the oldest forms of life that we have learned about. And here are some pictures of them. We've got these cyanobacteria. We used to call them blue-green algae. When I was growing up, that's what I learned because that's how old I am. And we didn't even have domains back when I was taught in grade school. We were taught just kingdoms was it. We weren't, my grade school was not taught about domains. I didn't learn about that until I was older. So the domain of prokaryotes, the domain of bacteria and archaea, sorry. those two domains, that's what these guys are. So these guys are going to be found there. Eukaryotes are plants and our fungi and all of those guys. Those are the little bitty guys. And if you put enough little bitty guys together, they can make a mat. Um, I'm hoping all of us are brushing our teeth. You're supposed to brush them in the morning, right? You're supposed to brush them before you go to bed. And when you're brushing your teeth, what you're brushing off is basically this stuff. When you have enough unicellular guys laying on top of each other, it forms a mat or a biofilm. And the plaque that you are, it's really gross, but when you brush your teeth, you're removing the biofilm on your mouth. Your teeth are covered in a biofilm. We have bacteria growing all over ourselves. Don't be alarmed. You are covered outside and inside in bacteria and fungi. Um, and they live there and they grow there and they're totally happy there. And a lot of times they're not harming you, and sometimes they are helping you. So you have bacteria everywhere. The ones on our teeth, some of these guys are fine, but some of these guys do harm us, right? Sometimes they can give us cavities. We can get dental caries from that bacterial mat on our mouth. So that's a form of a biofilm. Here is a picture of some of the oldest biofilms out there they're so old, they're fossilized. And these guys are called stromatolites. So we are always learning about new stuff. We are figuring out how things are changing and evolving. Science is always evolving. And it doesn't have to be, the natural sciences. It could be the tech, it could be tech science. We are always coming up with, different things, different software, newer versions of, cell phones or iPads or whatever. It's always changing and evolving. Things Other things that you will always hear about. You, whether you know it or not, are always surrounded by science. You will always hear about it on the news, whether it is some sort of foodborne illness. We talked about microbiology with the little bitty guys. So salmonella or E. coli can definitely cause food poisoning. So you may hear about a food illness outbreak in the news. You um, probably, hopefully, have heard about climate change, that it is most definitely happening. There is lots and lots and lots of evidence out there to show that it is happening. So that's another topic that's always out there in the news. Again, the newest tech gadget out there. Um, Science is it's out there. It's always changing. We're always studying and learning new things. I have a friend of mine, we're really close friends across the street. And he was telling me, that he's worried about the new COVID vaccine because we don't know. And I said, that's part of science, is that we we don't know all there is to know about the coronavirus, and we don't know everything about it. We don't. We're still learning. But through the scientific method, we have figured out what works and doesn't work. um, Not all of the details, but some basic details. We have figured out what works and doesn't work, and that has helped us produce new things like the COVID vaccination that's out right now. And there's two versions that are out right now at the time that I am filming this video. I'm sure it will be outdated, next month. But those scientific methods, the scientific breakthroughs that are happening is because of this. And so, although we don't know everything about everything, we are learning and proving what is true and what is not. So, we are going to go through those steps of scientific method in just a bit to learn about those. We mentioned this previously in the previous video when we talked about natural sciences or life sciences, and then physical sciences, things that are not alive. Chemistry is a physical science. Physics is a physical science. Astronomy, geology, none of those things are alive, right? They don't have the properties of life. So those are physical sciences. Life sciences, zoology, botany. Even if you went to zoology and you went further down, if you split up animals, zoology is the study of animals, you could break that down into mammalogy, studying mammals, me and you, or pets, the dogs or the cats that I have sitting here in my bedroom with me. Um, we could do mammalogy. Maybe you're a herpetologist. Maybe you are like the late great Steve Irwin or one of his kids that are still following his footsteps, and you really love reptiles and amphibians. Maybe you're a herpetologist, or you like birds. You're an ornithologist. You could be an ichthyologist, like our division chair in the school STEM. He loves fish. He's an ichthyologist. So even within zoology, there are branches and branches of those different things. But if you are studying living organisms, that is a natural or a life science. All right. How do we do the scientific method? First, we are going to just question and reason what's going on, right? We're studying our environment. Science is trying to study and make sense of our environment. So we're going to have two types of reasoning when we do this. We have inductive reasoning and we have deductive reasoning. So inductive reasoning is going to make a generalization um, that is going to take into account large numbers of data on specific observations using these two forms. So if we were talking about inductive reasoning and we are looking at a large quantity of data, and um, if I am, as an example, I could be, yeah, I could be Robert Hooke. And so Robert Hooke was looking at all of these different organisms. He was slicing up some cork and looking at it under a microscope, and he noticed that the cork had all of these, square shaped little things, rectangular shaped little structures inside, and he thought that was kind of cool. And then you look at the animals and you slice them really thin and you put them under the microscope, and you can see that they too have repeating structures in there under the microscope, and you start to think, well, if I've looked at all of these things, I looked at all these plants and I looked at all these animals, and I keep seeing these same kind of basic structures, and I'm going to call those things cells. So based on this inductive reasoning from this large massive quantity of data that I've looked at, I am going to make this generalization that everything that is alive, all living organisms, are made from cells. I got to that from my large quantity of data that I looked at, and my inductive reasoning is going to come up with this hypothesis that all living organisms are made from cells based on my large selection of data. What is data? Data can come in two types. So there is qualitative and there is quantitative. I'm actually taking classes in those for my doctorate in education, which you would not think that you would need to take statistics on qualitative and quantitative data analysis for a doctorate in education, but you do. So, in my qualitative data analysis class, that data is descriptive. So, an example of descriptive data would be I am going to interview somebody and record that interview. I will ask a person questions and I will record down their responses. And that data is qualitative data. It's good stuff. And if I interviewed um, patients that received the COVID vaccination and I interviewed them and asked them what their side effects were, and they described those side effects to me, that would be qualitative data. Well, I had a little irritation at the site, but everything else seems to be fine. That's data. It's not necessarily hardcore numbers, but it's still data. So that's qualitative data. Quantitative data is usually what I think of in my scientific mind. That's what go towards automatically is quantitative data, numerical data, numbers. Can I crunch the numbers and get to something and see if something is statistically significant or not? So sometimes you will have a mixed method study. Sometimes you will take down both quantitative and qualitative data. But there are two types of data, and I want you to know that can be both. You can take down numbers as quantitative and analyze those. All of the um, fever, all incoming COVID patients and what their fever was upon entrance into the hospital. How many people had a fever of 99? How many patients had a fever of 100 degrees? That is quantitative data. But it is also if you took down their symptoms, um, w- what, you know, how did they describe how they were feeling? That'd be qualitative data. So based on information, we can reason out some hypotheses. So if we are using inductive reasoning, again, that is coming from a large pool of data. You're going to make a generalization based on that large pool of data. And an example would be all living organisms come from cells because I looked at a ton of cells under a microscope and I think that everything that's alive has cells. That could be my example for inductive reasoning. Deductive reasoning is going to use a general principle or law. So laws like the law of gravity or a commonly accepted principle. We are going to use that to predict something, to predict a result. So we're going to use the data to explain science. So, our example, if our local climate is becoming warmer, so we're noticing the temperature is rising in our local area. If the local climate is becoming warmer, that quantitative data is showing that the climate is changing and becoming warmer, then based on our principle that every organism is evolved to the environment that it's at. Remember, we said that those traits are going to help it be perfect for its environment. And if it doesn't have the traits for its environment, it's not going to live very long. So, based on that, every organism is in the environment and they're evolved for that environment. So, if the environment changes, if our local climate is becoming warmer, then the distribution of plants and animals will change too. So, based on that knowledge, that principle or law that animals and plants, the organisms that are there because they evolved to that specific type of geography and climate and temperature. If the climate changes, then the distribution of plants and animals are going to change. That is an example of deductive reasoning based on some sort of knowledge that you've got there, some principles, some law, some foundational knowledge. You are going to predict something to happen. So, if the local climate is becoming warmer, then the distribution of the plants and animals will change. So, you may have to know on the test an example of these, or maybe you'll be given an example and you'll need to know, are we talking about deductive or inductive reasoning? Or maybe you'll be given a question about data. And is that data an example of qualitative data or quantitative data? Um, pathways of discovery, pathways of scientific inquiry. The scientific inquiry could be descriptive or hypothesis. Our descriptive science or discovery science is going to discover, it's going to observe, take notes, write down, and explore different environments, different conditions, and write all of that descriptive information down. I chose Glacier National Park as my background in this video. It's one of my favorite national parks. It's where my parents retired when I went to college. My parents moved to Montana from Alabama, and they lived like 40 minutes from Glacier National Park. So we went there. I took my kids there last year on a road trip. Husband, my three kids, the two dogs, the two cats. It was like it's like a clown car and a trailer that we hauled across the country last year. And we went to Glacier as one of our stops. So I took this picture there last year. And if I was going into Glacier for the first time ever, I would write down all of this descriptive data. You know, what is the sun like? What is the temperature like in Glacier National Park? What plant life am I seeing? This picture is taken from a trail called the Highline Trail. What plant life do I see in the Highline Trail versus what plant life do I see way down um, in the bottom near the river that's going down into Flathead Lake or Lake McDonald? What life do I see down there? What animals do I see? What plants do I see? How are the glaciers doing? Are they receding? They are, by the way. The glaciers are receding. Um, where is the glacier right now on this day? If I come back in 20 more years, where is the glacier on that day? Isn't even still around. Let's hope it is. That's all descriptive science. observing, exploring, discovering knowledge. Then there is hypothesis based, and hypothesis based is going to be you start off with a question. You want to solve something. I want to cure cancer. I lost my brother-in-law to testicular cancer two a little under 2 years ago. We lost him. And so maybe that's my goal in life. Maybe that's my hypothesis based science that I want to cure testicular cancer. And that's going to be my goal. That's my aim. That's my problem. And I'm going to solve it. So that is another type of pathway that you could choose. So there's really kind of a descriptive discovery pathway, and there is a focused hypothesis based, problem based type of pathway. Scientific method. I want us to know this. Scientific method was developed by a guy named Sir Francis Bacon. And we use the scientific method to be able to test and prove things. We need to be able to come up with a hypothesis. Here's our Sir Francis Bacon. Here's our guy. You came up with this method, and we all learned this, right, in grade school. We know this. But we're going to go through it one more time because remember, we need to know these steps. And this is one of the founding principles of science is the scientific method. It is why when we have this overabundance of information out there on the internet, that I want y'all to understand what is real and what is not. I need y'all to be able to understand the scientific method. I need you to understand what is legitimate research and what is misinformation out there, because there is so much misinformation out there and false claims. And I want y'all to be able to understand what is true and what is not. Get to the heart of something and discover, is this legitimate information? Is this study really true? Or is, I saw this article that said that the COVID vaccine makes everybody infertile. Um, Look, find out, discover, research. So we are going to talk about what makes something a legitimate claim. So the scientific method, let's start off with our hypothesis. And our hypothesis is our educated guess. We are making an educated guess because if we are doing an experiment, it needs to be testable. We need to have an educated guess, and if then is usually how it's written up. If um, I eat healthy foods every day, then I should live a long and healthy life. Some sort of testable hypothesis. If I flick the switch on the wall in my room here, then I expect the light to turn on. That is my educated guess. That is my hypothesis, and it needs to be testable. You cannot have a hypothesis on something that you cannot test. I cannot test somebody if they say, how can you test if somebody says, I believe in ghosts? Okay. I mean, that's great. And you are entitled to whatever beliefs you want, but I cannot test that, right? I cannot test somebody's statement that they say they believe in ghosts or, the boogeyman or whatever. Um, that's you can say that all you want, but you can't test it. It's not provable. So scientific inquiry needs to be testable. It needs to be provable. So you're going to come up with an educated guess, and that is your hypothesis. And then that hypothesis, if we run through the scientific method steps, eventually we are going to go through all of these steps. So we have this observation that there is a light switch on the wall. And then we're going to ask a question. I wonder if that light switch on the wall controls the light switch up above, or maybe it controls the lamp next to me on the table. So I'm going to ask a question, and I think if I switch, if I flick this switch of the light switch, then it's going to turn on the overhead light. And that's going to be my question. and I've formed a hypothesis. I have written it as my if then hypothesis, my educated guess. If I flick the switch on the wall, then the overhead light will turn on. That is going to make my prediction based on the hypothesis, and then I'm going to perform an experiment. So, what's my experiment? Well, my experiment in this case is going to be I'm going to walk over to that wall switch and I'm going to flip the switch and I'm going to document what happens when I flicked the switch. And so I'm going to document that data, I'm going to write it down, and I'm going to analyze the results. When I flicked the switch, did the overhead light turn on or did it not? Did it not turn on? Was it not that light? Did it turn on the lamp? If it turned on the lamp, that's still data. It doesn't prove my hypothesis, it disproves my hypothesis. And if it turned on the lamp, then my hypothesis would be incorrect and I would try again. If I flicked the switch and I wrote down my data and it turns out that my data was that the overhead light turned on, then my hypothesis is correct. And I would write up those reports and those results. Even if you don't get your hypothesis proven, right, you still want to write up those results. There is some quote out there about the light bulb, and I it was talking about how he didn't discover a way to create the light bulb. He discovered like 1,000 other ways to not create a light bulb. Um, because again, all of those errors, all of those educated guesses that didn't come true, that still produce data, and it's data that is important. Good data, data that didn't support your that didn't prove your hypothesis to be correct is still good data. All data is good data. Write it down and report the results. And if you need to try again with your new hypothesis, alter your hypothesis and try again. But when you follow the steps of the scientific method, you should be able to, have your observation, come up with your hypothesis, do an experiment to test your hypothesis, collect your data, and determine if that data was correct and report the results. If you come up with a hypothesis and you prove this thing, then somebody else is going to come along and be like, nope, I think you're full of it. I'm going to flick that switch. And I don't think that switch covers the overhead light. I think it covers the light outside. It's the floodlight. It's not the overhead light. So I think you're full of it. And I'm going to follow the steps of the scientific method. I'm going to follow all of your methodology that you used and the data that's looking at your data collection. I think you're wrong. So I'm going to do the same exact experiment and I'm going to see if it really does turn on the overhead light or if it turns on the floodlight outside. I think you're full of it and I'm going to test it. So somebody else comes along and tests it. And if they follow the steps of the scientific method that you wrote down and your results in your paper, and they're going to find that either it works for them or it doesn't work for them. And if enough people determine that this thing really keeps happening, then it's true, right? If enough people are able to recreate an experiment over and over and over again and get the same results, it's going to become a theory or a law because it's been testable. That is the glorious part about science is that anybody can come along and make a statement. I can turn lead into gold. But if somebody cannot follow those same steps and recreate it, then your study is baloney. Somebody has to come along and be able to reproduce your results. Can somebody do that? Is this happening? Are you getting the same results every time? Then that hypothesis is probably true. If somebody else is doing this and they are claiming that, they've got an outlandish claim, then I want you as, a scientific inquiry person, an enlightened student now, to question if you see crazy claims out there. Look at where this is coming from. Is this coming from a scientific study? Who performed the study? How did they perform the study? Who funded the study? Track the money. That's another really good, important factor. Because if you see a whole bunch of studies that are claiming that climate change is happening, here's the evidence for climate change, and there's one random study that says, nope, climate change is a whole bunch of baloney. It's not happening. And that study happens to be by company that will be impacted if people start becoming aware of climate change, then that, you should call into question the results from a study funded by, I don't know, a petrochemical company, something like that. Who is funding the study? I want y'all to always be questioning. That is one of the main takeaways I want you to get from this class as a whole in biology is question your information that's coming to you in this information age. Question the source. Is this a legitimate scientific study? Did they follow the steps of the scientific method? Um, who published it? We're going to get to that in just a little bit. So these are the steps of the scientific method. You need to know the steps of the scientific method. You probably already know these from grade school again, but just know your steps. We may have questions about variables. So variables, we are going to have independent variables and dependent variables when we perform experiments. So an independent variable is something, if you are an independent person, you are totally cool with being by yourself. You can stand alone and you are not changed by other things. So examples of independent variables. Age. How old are you? It does not matter. Age is not affected by, how much sleep you got or what you ate for breakfast. Um, age is an independent variable. It is not impacted by other things. Dependent variables, on the other hand, though, they change depending on different factors. So an example of a dependent variable is test scores. Test scores can change, right? You can completely ace a test or you might completely bomb a test. And that can be from a number of different things, right? What are all the different things that could cause you to perform poorly on a test? I can tell you right now, not preparing for the test will cause you to perform poorly in this class. You are going to need to study for this class. So if you didn't study, that's a variable that impacts your test scores. I only was able to give myself 5 minutes to look over my notes before I took the test. Going to impact your test scores. Maybe you didn't get a whole lot of sleep the night before the test and you came in and you had to do like the ACT and you didn't sleep all night. That might impact your test scores. All sorts of things can impact your test scores. Maybe you got a great night's sleep, you got a good breakfast, and you studied, and you rocked your test. And you got, one of the best grades in the class. Lots of different things can impact your test scores. Test scores are dependent variables. They are dependent on the different things, on the sleep, on the food, on the preparation, how many hours of study time went into that test. So test scores are dependent variables, while something that stands alone, like age, is an independent variable. Okay, so if you were to plug them into this sentence with your dependent variable, your independent variable, and your dependent variable. So if you said your independent variable causes a change in the dependent variable, but it's not possible that the dependent variable could cause a change in the independent variable. So if we said age can cause a change in test scores. There we go. Age causes a change in test scores. Maybe you are really, really young and you are maybe you're in first grade and you are given a test at the level of, senior in high school. Age would cause a change in test scores, but it's not possible that test scores would cause a change in age. It doesn't matter if you fail the ACT, that's not going to change your age. It's not going to make you younger because of your test scores. So if you can remember that phrase, that the independent variable can cause a change in the dependent variables, but those dependent variables do not cause a change in the independent variable. Hopefully that can help you differentiate between the two. So you will need to know the difference between dependent variables and independent variables. Basic and applied science are kind of what we talked about previously when we talked about descriptive science and hypothetical science. Our basic or our pure science, you just want to see what's out there. You want to expand the knowledge. You want to go and explore and see and do the things. You want to see what's on the other side of the moon just because you can. I just want to get there. I want to see what's out in space just to see what's out in space. That is pure basic knowledge, basic science there. And then there is applied science that we talked about, where I said that maybe my goal is to cure testicular cancer because I lost my brother in law to that. And I don't want to see anybody else lose their brother in law to testicular cancer. And that would be applied science. I have a goal. I am trying to use the science to solve the real world problem of cancer. Maybe your goal is to improve upon the COVID vaccine because now that we've got that out there, maybe we need um, some other kind of goal. I am sure there will be some other new great goal out there for you to do something. Maybe you want to save an endangered species and try and help that. That would be a goal. So it is an applied science. Sometimes it's a blend. Sometimes a study can be both. It may be basic science and applied science. An example of that is the Human Genome Project. So this Human Genome Project was started back in 90, yeah, 1990 is when this started, and it took them 13 years. And so they just, wanted to see, we learned about DNA. We figured out its structure about 1950s, what its basic structure is. And then we've been learning about it ever since. And we know, where it's at, but we didn't know exactly what it did, where those chromosomes, what genes were on what chromosomes. So we really just kind of wanted to look into that and see more about that. So the Human Genome Project is a bit of both. It is both basic science and applied science. So with the Human Genome Project, we wanted to map out the genes, what genes are on what chromosomes, and then what are the genes coding for. So now we can try and use that genetic information for all of these applied goals. You can see here on this picture, we can see all of the chromosomal pairs. We are, um, as humans, we are going to have two sets of chromosomes. We are going to have a set from mom and a set from dad, and we're going to say this chromosome here is going to have all the genes that code for hair color or whatever over here. And this is dad's chromosome. And then this is mom's chromosome, and it has all the genes that code for hair color. So when we put these guys together, they're called a homologous pair. And we have 23 of these in our cells. So we took all of those 23 pairs of chromosomes and we put them together as part of this genome project. And we mapped out every chromosome, what the DNA is on that chromosome, and what does it code for. And so we can see here on chromosome 17 that has the breast cancer gene on there. If you have the breast cancer gene, does that mean that gene will express and you are bound and determined, you are fated to have breast cancer? No, doesn't mean that you're bound and determined to get breast cancer. Just because you have the gene for it does not mean that it will express. That's the same for all of these other genes. Just because you have the gene for prostate cancer does not mean that gene is definitely going to express and you are always going to have prostate cancer. Something turns on the gene and causes it to express to then become the overgrown growth that is cancer. So all of these different chromosomes and the disorders associated with those chromosomes. This is not only pure science, just to check out the chromosomes and see what the DNA is on there, but it is applied science. So now we can see what that DNA codes for, what it expresses, what happens when it becomes expressed. This is the Human Genome Project. So super cool, amazing study. Took 13 years to map this out. And this was in the 90s where DNA technology was really long, very intensive, super expensive. And now we can map out a genome in like no time using computers and newer technology. Technology in, again, biotech, using computers and biological knowledge is still very much huge. It is still a booming industry in biotech. Okay, so back to science and why I want you to understand the difference between somebody's study that they mentioned in a blog that they really just, gathered up these anecdotal notes and then came through with this idea of, you know, um, whatever. I need y'all to be able to look at this and be able to critically analyze claims. Like it's still out there, but I guess about 15 years ago is when I first started hearing about it, with the anti-vax, and that the anti-vaxxers, the people that are against vaccinations, they really started to, I guess, make the news that it was really more prominent when a study came out by a guy, a doctor out there who claimed that the vaccinations are what caused autism, that autism was a result of vaccines and what was in the vaccines. And so that scared a lot of people. But it turns out that that study wasn't a legitimate study. It was not a good study. It was full of errors. It was incorrect. But that one study from that one scientist set off a huge wave of people being concerned that vaccinations are going to cause a whole bunch of terrible things to people. So I want y'all to be able to critically think about where the information is coming from. Um, and is it a peer reviewed study? Is it coming from a blog? Where is this coming from? Because not vaccinating the population has definitely impacted a lot of people, based on the information from that guy's study that turns out wasn't even a legitimate study. So, how is it a legitimate study? Well, one of the ways that you can determine if a study is a legitimate study is it peer reviewed? What does peer reviewed mean? Peer review is your peers are your friends, right? So if you are looking at a study, is it in a peer reviewed journal? Is it reviewed by a group of peers? So I could come up with a study um, with my feral cats. That's what my master's was on feral cats. And so I studied trap, neuter, return, which is a method of population control for feral cats. Well, you trap the cats, and instead of trapping them and killing them, which usually is what happens, you trap them and you sterilize them and you vaccinate them, and then you put them back out in the population. So, if I had been able to publish my study, I would have published my study in a peer-reviewed journal. It's not published. It's out there on it's out there, but it's not published in a peer-reviewed journal. And that is so if somebody saw my thesis, it wouldn't be as strong as if I had published it in a peer-reviewed journal. So if I had been able to publish my work in, say, the Journal of Veterinary Medicine, JAVMA, that would have been good. Or if I had been able to publish my study in the Journal of Wildlife Medicine, Journal of Wildlife Management, the JWM, that is a peer-reviewed journal. And what that means is a bunch of, if I was going to the Wildlife Journal of Wildlife Management, for example, I would submit this to the journal, and a bunch of wildlife biologists would look at my study and they would look at the way I did it, because when you write down your study, you write down your introduction, your materials and methods, why you did what you did, how you did what you did, what data did you collect, how did you analyze your data, and then what did you do for your results and your discussion. They look at all of that, and they may come back to me and be like, you know what, you only studied a small number of cats, and the deduction that you made from this small number of cats, there's no way that you could make that conclusion. Or maybe they came back and they were like, you know what? I think that you analyzed your data wrong and you should use a different method of statistical analysis. You need to use this method instead of this other form of method, and then come back to us with this new rewritten using this new analysis method. And then, we'll look at it again. So these are these are your peers looking at your study and basically tearing it apart um, before it gets published. So in this day and age, you can put out a whole bunch of stuff out online and claim that it's true and it's a legit study, and it may not be. So look and see where this information is coming from. Is it coming from a peer reviewed source? Is it coming from a published journal? Is it coming from, something that you could search for in Galileo, Science Daily, any of those peer reviewed publications? If I published it on the Kim Subach blog and then claim that it's a legitimate study, that's not the same. I'm going to take the peer reviewed gold standard article well before I take some Joe Schmo blog over here. So I want y'all to be able to understand this. I want y'all to be able to pick and choose what is legit research and what is not. And in the scientific world, peer reviewed journals are the way to go. A peer reviewed publication. Maybe this person did a conference or a meeting. Maybe they did a TED talk. Those are also usually experts in their field if you are seeing somebody at something like this. But this peer reviewed is the gold standard for scientific work. Has this study been published in a peer reviewed journal, whether that is a old fashioned paper journal that comes out once a month or an online peer reviewed journal? Most of the journals now are all online. Um, is it a peer reviewed source? Is it a random blog? Um, if I had been able to publish, I would have mine out there in a peer reviewed source. My husband, his master's is out there and he's published. So he has a leg up on me. He's got two publications out of his master's study, and I didn't get any out of mine. But that again is the gold standard, is a peer reviewed source. I want y'all to be able to determine who's doing it, where did you see this information, um, where was it published, who is the author, who funded the study. All of these are really, really important things to determine and figure out. Think critically about the information that is coming to you. That's my big thing from me to you that I want you to take out of this out of this class. If nothing else, please be able to sort through information and determine what is, quite frankly, crap, and what is what is real legit science that has been tested over and over again and coming to the same conclusions. That is true scientific work. Stuff that has been tested, it has been reviewed by peers. Um, that's the good stuff. And I want y'all to be able to know that. So, this is chapter one. This is the last of the stuff from chapter one. Please go through, know your chapter outlines, read your textbook, study, do the study guides, and let me know if you have questions about any of this.